Okay. Okay. All right. Hello, everyone. So let me see. So firstly, I hope So now that 3 weeks of the course is now uploaded, I hope that you've been following the lectures and that you've been enjoying the material that we've covered so far, which is basically until diffusion processes in cells. And mm, basically diffusion, FRAP, cell signaling, and so on. So things to do with diffusive motion inside a cellular environment. Next, I think we're going to start with the fluid dynamics of cells. Mm, how the local environment inside the cell, which is like a very viscous, very thick fluid, how that is going to affect second, how that is going to affect biological processes inside the cell and so on. Right. So while some of you have asked some questions on this response sheet that was shared, so I will address those questions. In the meantime, whoever is present, if you have any additional things you would like me to discuss, if you would um, like me to clarify certain aspects of the lectures, or if you um, you know, if there's some very even general questions that you want to ask, whether about the course or whether about basically biological physics in general, please feel free to type in the chat box, and I'll try to answer as best as I can. Okay. Now, so I'll take some of the general questions that came up in this Google Sheet first, and so one of the things that someone has asked is that what physics topics sort of do you what kind of physics is most useful for understanding biological systems, and that does not have a very precise answer basically. So depending, so that's sort of the beauty of biological physics. That's also the challenge of biological physics, and that's also the opportunity in some sense. See, biology, you know, is a very broad set of phenomena, right? You have plant biology, you have animal biology. Inside the cell, you have various processes that are going on simultaneously. Depending on which of these processes you're interested in trying to understand, you need a different set of toolboxes, right? So, for example, um, we have done is basically diffusion. So that's the first topic that we have covered, which is diffusion, right? Um, Now, diffusion, you might imagine, is a very basic sort of physics concept. You write down the diffusion equation, you try to solve the diffusion equation, and then you apply it to a biological context, like we saw. We saw this diffusion equation being applied to let us say, the cell signaling problem, right, which is a biological problem. Um, so that's one sort of toolbox. Now, depending on, for example, so therefore, depending on what question you're interested in, you can use different techniques that are best suited for understanding that biological system, or for answering those questions that has been asked. So in this course, what I generally do is that I focus more on statistical, so biological questions that can be broadly answered within the framework of statistical mechanics. Okay? That can be broadly answered within the framework of statistical mechanics, thermodynamics, basically. Okay. Also, to a certain extent, non-equilibrium processes, so basically rate equations in the language of rate equations, and mm, a little bit of so stochastic processes. Stochastic processes are things, where processes happen so there is some randomness associated with these processes, which is often true in biology. But for for example, there could be various different lenses through which you can look at some biological problems. For example, you could look at it using a toolbox of, let us say, mechanics, okay, which would come under, let's say, biomechanics. So, for example, just to give you one concrete, so I do not, in this course, for example, I um, don't there are some problems where I do a little bit of mechanics, but I don't focus a lot on the mechanics aspect of it. On the other hand, somebody else might focus a lot more on the mechanics aspect and not so much on the statistical physics rate equations aspect. So there's a very diverse set of topics basically that you can use. So just to give an example of this mechanics, since I was talking about that, you could imagine. Let us say, a cell dividing. Let us say a cell. I have a cell over here, and this cell is going to divide. Right. So this cell is going to become two cells. Right? This cell is going to become two cells. And how that starts is that basically you have this cell wall that sort of starts to septum, basically, that starts to form in the middle, and then that sort of progresses more and more until it divides of your original cell into two parts, and then the cell splits into two, and you have cell division. So among other factors, so of course there are genes and so on that play a role, but among other factors, often what is important is what sort of forces are being generated by this septum. And so then, if you were looking at a problem like this, you would need the toolbox of mechanics. Uh, so that's one sort of thing that you could be looking at. Then of course there's of course the physics of fluid flows, which is what we talk about a little bit, in the next in the lectures of the next weeks and so on. So fluid flows and low Reynolds numbers. So here, as you'll see, what we'll do is that we'll start from the Navier-Stokes equation, low Reynolds numbers and so on. So what we'll do is that we'll start from the Navier-Stokes equations, and we'll see in what limit these Navier-Stokes equations are relevant to biology. And then we'll try to understand certain biological phenomenon in those limits, which is basically this low Reynolds number regimes. So there is no. So the good thing about doing this sort of physics of biological systems is that there is no, depending on what is your strength in physics or in maths, you can pick a biological problem where you can apply those ideas. So in this course, I focus on certain things because that's what basically I'm interested in. That's that's the sort of research that I do. But if you're more broadly interested in biological physics, and you have a certain set of toolboxes that you're more familiar with, then you can find a biological problem where you can use those toolboxes in order to answer questions that are of interest to a biologist. So there is no unique set of toolboxes. Depending on what is your interest and what is your strength, you can actually find biological problems where you can apply those toolboxes in order to answer very relevant biological questions. Okay, so that's one thing. All right, now somebody has asked about some background learning, background notes. About the basic mathematics and statistical parts. So what I'll do, so basically background reading. Okay, so let me come to this next thing, which is background material. So I generally tell people who take this course that ideally it is best if you have some sort of knowledge of statistical mechanics, because that is equilibrium statistical mechanics is probably sufficient. So because that is something that I keep coming back to throughout the course. So equilibrium statistical mechanics, and I'll use it more and more as the course sort of progresses. Um, So basically, what that means is that how does one calculate thermodynamic averages? Basically, what is energy? Okay, what is energy? What is entropy? What is entropy? What is free energy? So on. So we'll use all of these concepts as we go forward to look at biological systems. So if you don't have a very good understanding of these concepts, you might find certain topics a little difficult to follow. Um, what I could do, I have a set of notes on equilibrium statistical mechanics that are relevant. So what I could do is that I could point you to, and I have those notes on the web. So I could point you to these notes, and you can take a look at these notes. So basically, but ideally you should know a little bit of this on your own, and then you can use these notes that I can upload to sort of refresh your memory, or to sort of refresh these concepts of what's energy, what is entropy, what is free energy, and so on. How does one count microstates, macrostates? How does one calculate partition functions? How does one calculate thermodynamic averages, and so on. So, uh, if it's a question of sort of revising some of these concepts, then please feel free to use my notes. If you have never come across these concepts before, if you do not know what entropy is, um, you can still study from my notes. But what I mean is that you'll basically then have to do the work of two courses in one, right? Because you'll have to understand a certain level of equilibrium statistical mechanics before you can appreciate some of these calculations that I'll do in the weeks ahead. Basically, some parts are standalone. So, for example, this diffusion, I don't think you needed a lot of background. A little bit of mathematical knowledge, of course. You need to know how to solve differential equations. And so on. But beyond that, you don't need a lot of background. Similarly, the next part, which is this fluid dynamics, Navier-Stokes equations, and load and all numbers, again, I sort of try to build it from a point where you do not need a lot of background knowledge. But the next part, where I deal with this energy considerations, energy, entropy, and so on, it would be helpful if you understand, you have a basic understanding at least of what these concepts mean. Uh, Um, so that's what I'll do. I'll upload. So I'll upload a set of notes on this equilibrium. So or rather, I have these uploaded. I'll point you to where you can download these notes. Notes on this. Notes on these. Um, Other things. So, like for example, if there is a particular toolbox that I use. That you feel you do not really understand, and that's perfectly fine. Um, Please post in the forum, and I'll try to point you to a resource, some textbook or some online resource where you can read it up. Okay, so there's no harm if you do not, if you it's hard for me to give a very general link, but if you tell me that, okay, so this topic you solved the differential equations using this technique, and that I do not really understand, then I'll try to, I'll try to upload whatever notes are um, mm, whatever notes I think are suitable to help you understand that. So that's number two. All right. So that was another question. All right. Now coming to, so that's mostly about sort of background information and so on. Coming to the next question, somebody has asked about how genetic information is encrypted. Genetic information is encrypted by the DNA. I think what you mean, how is genetic information encoded by the DNA? And so I think I explained a little in the lectures, but I can go over this a little bit again. Okay. Okay. Um, it's actually interesting. It's good to know. I mean, it's okay. So how is genetic information encoded in DNA? Okay. I assume this is what you wanted to ask. If you mean something else, so I'll explain this. If this does not precisely answer your query, again, feel free to post on the forum or send me an email, and I'll try to answer again. Okay. So here's the thing, right? So what we discussed is that on DNA, you have this string of 4 alphabets, which are your nucleotides A, T, C, and G, right? Adenine, thymine, cytosine, guanine. Okay, so this A, T, C, G are the letters of the alphabet that make up your genetic code. Now, I have my double-stranded DNA. I have, let's say for here, I have my double-stranded DNA. Okay, on which I have this sequence. So let's say A, C, G, C, T, T, A, and so on. Okay, on one strand, and on the other strand, then I have the complementary pair. So T, T, C, G, A, T, so on. So here I have, so basically I have this sequence of nucleotides that comprises my genetic code. So this is so each of this is a base pair. Each of this is one base pair, and then my human genome, for example, is roughly like 10 to the power of 6 such base pairs. Okay. It's a lot of base pairs. So it's a lot of information that is coded inside on your DNA, and this is present in every single cell of your body. Okay. So basically, you have 10 to the power of, so I have 1, 2, 3, 4, 5, 6, 7 such letters over here that I have shown. In the human genome, you'll have of the order of 10 to the power of 6 such letters. Okay. So this whole sequence, this whole string of letters, A, T, C, G, A, and so on, that comprises my genetic code. Okay, so let me write it down. Let me write like something. So instead of writing it on the background, let me just write over here. So A, G, and I'll write only of one strand. The other strand is just the complementary A of this. So A, G, C, T, T, A, G, G, C, C, T, A, and so on. Okay. And this continues for 10 to the power of 6, actually. So it's a huge amount of information. Now, the way the information is stored is that, so this whole thing that I have, this 10 to the power of 6 base pairs in the case of humans, comprises my genetic code, comprises my full genetic code. Now, not all of this codes for proteins. Okay. So only certain segments of this 10 to the power of 6 base pairs will actually code for proteins. Okay. So, and certain other parts will not. So certain other parts will have maybe, we do not really actually completely even understand what are the roles or functionalities of all of this remaining base pairs. Some we do understand. I mean, over the last 20, 30 years, we've understood more and more as to what roles these parts which do not code for proteins, what roles those parts play. But even now, there are n number of questions that we do not really know the answer to. All right. Now, to give a very simple example, let us say I have this string of alphabets over here, and let's say only, let me see, 1, 2, 3, 4, 5, 6. So let's say over here is something that codes for a protein. Okay. The remaining parts do not. So the way it codes, so again, we have discussed this. So you have two processes. One is transcription. Where this genetic information is copied onto RNA. And then there is translation. Where this genetic information on the RNA is translated in order to produce proteins. Now, the way proteins, amino acids, so proteins are comprised of amino acids, and the way amino acids are produced is that three of these nucleotides, right? Three of these nucleotides will code for one amino acid, so it will code for amino acid 1. Then the next three nucleotides will code for another amino acid, so let's say amino acid 2. And so the genetic information is read in triplets, right? So here TTA is one triplet, which will produce one amino acid. GGC is the next triplet that will produce the next amino acid, and so on. And this way it will go on until it comes to a certain amino acid which tells the cell to stop, okay, to stop reading this genetic code, and that's when it will stop producing these amino acids. So these parts of the DNA, or these nucleotides which code for proteins, are technically what is called as genes. So these are your genes. Okay? The protein coding parts of your genome. The genome is this whole system, this whole string of 10 to the power of 6 base pairs is your whole entire genome. Now, those segments which actually code for proteins are called the genes. Okay? And those segments which do not code for, so these parts over here, this over here is a different color. So these nucleotides over here, or these nucleotides over here, they might have different roles. For example, they might have regulatory roles. Now, what sort of regulatory roles am I thinking about? So let's say here is my whole DNA, right? Let's imagine that this is a double-stranded DNA, and then there are parts of this DNA. There are sequences along this DNA which code for proteins. So here are these sequences. Okay. So here is a sequence which codes for gene. Here's gene 1, which codes for protein 1. Here is gene 2, which codes for protein 2. Here is gene 3, which codes for protein 3, and so on. Okay. So there are lots and lots of nucleotides here. A, C, D, 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 lots and lots of lines. Okay. So these can be fairly long strings. And then there are other nucleotides over here, A, T, G, C, which do not ostensibly code for proteins, but they still have a role to play. And what sort of roles could they have? So they could have a regulatory role. And by regulatory role, what I mean is that, for example, they could regulate when these genes are expressed, okay, or to what levels these genes are expressed. Now, what do I mean by that? What I mean is, so this gene is first, through the process of transcription, gets converted to RNA, and then through the process of translation, it gets converted to protein. So the ultimate output of this, is basically that you produce some protein. Okay, you produce protein 2. Okay. This gene 2 produces protein 2. Now, just because there is a gene that codes for this protein 2 does not mean that this protein 2 is being produced continuously by the cell. No, this protein 2 is produced by the cell as and when there is a requirement for the cell for this protein. Okay, so when this protein is required, the cell produces this protein. When this protein is not required, the cell stops the production of this protein. Now, but remember, this gene itself is not undergoing any change. This gene is, it's just a static sequence of nucleotides, A, T, C, G, whatever. So that itself is not undergoing any change. But the time behavior, the expression profile, which we call as the expression profile, the expression profile of this gene is undergoing a change with time. At certain times, this gene is being read and the corresponding protein is being produced. At other times, this gene is silent and the corresponding protein is not being produced. What determines whether this protein is going to be produced or not is basically what I mean by this regulation. Okay? So whether this gene will be turned on or it will be turned off is a question of regulation, and that can be achieved by interactions of this gene with other segments of your genome, with nucleotides over here or nucleotides over here. So maybe some protein comes and binds to this nucleotide over here, and by virtue of this binding, it tells this gene that, okay, now I need you to produce your protein, so please from this off state, please convert to the on state and start producing a protein, the protein that you're supposed to produce. And then maybe, when this protein is no longer required, this protein that bound to this and, told it to start producing, that comes off. So that dissociates from the DNA. And then again, this gene goes from the on state to the off state. So there is a lot of regulation that is involved. This is just one sort. There are more complicated processes that happen. I don't want to go into the complications. You can, of course, look up how gene regulation happens and so on. But the thing is this, that so there is this whole genome, which is this entire string of base pairs that you have, that every cell of your body has. Only certain segments of these actually code for proteins. And those segments, the protein coding segments, are what I call as my genes. The remaining could have different roles, and we do not really understand entirely what all of those roles might be. But one role is definitely, as I explained, a regulatory role. It could instruct the cells when to turn on a gene, when to turn off a gene, so that the corresponding protein can be produced at the correct time and to the correct desired level. So that's one example of a regulatory role. So it's a very complicated machinery. Your genome is static, but the expression profile of this genome is highly dynamic. And it also depends, of course, on cell type to cell type, right? The proteins that are produced in your skin cells are different from the proteins that are produced in your heart cells, even though the genome in your skin cell is the same as the genome in your heart cell. It is whatever genome that makes you, your own unique genome. So the genome is the same, but the expression profile, so what proteins are being produced, is completely different depending on the cell type that you're interested in. And even in the single different cell types, even if you were to consider a skin cell, it's not that the same set of proteins are being produced to the same level at all points of time. Again, depending on whether the cell is in interphase or the cell is in metaphase and so on. So depending on the cell cycle, you could have different proteins being produced and so on and so forth. So therefore, I suppose that what I want to express is that the way that information is encoded in DNA is in some sense simple, but at the same time very complex. So if you just think of the information as this this string of letters, that's a static thing. I mean, that is whatever is your genome. But the way it is expressed and the way that becomes life as such, that is a very complex process, and that's something that still trying to understand how all of these regulatory processes play a role. How proteins get produced to exactly the correct level that is required for the correct functioning of the cell. And that's a very complicated thing, and that's something that people still study. Okay. So I think that's about what was asked in the response sheet. Now let me see if we have some further questions. All right. Kriti has asked that in case of biomolecules of DNA, how does entropy change as DNA supercoils to form a highly condensed structure? Okay, very nice question. So we'll, of course, thanks for asking. I'll explain a little bit. But we'll also deal with this when we come to these sections on biopolymers. So I have, sorry, let me change the pen color back to. So when I come to this segment on biopolymers, biopolymers as in DNA, biopolymers as in RNA, biopolymers as in microtubules, so basically microtubules or actin, or even proteins, actually, proteins or biopolymers. So whenever you have some sort of a repeating subunit, that's what it's called, a polymer. So I spend, I suppose, a couple of weeks on biopolymers. So right now, we are doing low Reynolds number regime inside the cell. After that, we'll start a little bit of equilibrium physics, how we can apply concepts of equilibrium statistical mechanics in biological systems, and then we'll transition from there into applying these concepts of energy and entropy, these equilibrium concepts, to this study of biopolymers like DNA and RNA and so on. So when I come to that, I'll explicitly show you by doing some calculations as to how you can calculate entropy of these biopolymers in different contexts. Okay, so using very simple sort of counting examples. So when things are folded, nicely folded, and so on, I'll try to show you how you can count the entropy analytically, basically, and how to apply that to these biological systems. But that's okay. So let me give you a very short answer for now. So here's the thing. So here I have my polymer. So I have my DNA, which is a polymer. Now let's say it exists in a state like this, which is a sort of open state. Okay? Versus, let us say, it exists in a state where it is highly supercoiled. So it's very condensed state. Okay? So something like this. Okay. So here's open state of DNA. This is, imagine that this polymer that I have drawn is DNA. Okay. Imagine that this is a DNA double helix. So here is an open state of DNA. Here is a sort of very condensed state of DNA. Now, what is entropy? Entropy is basically how much, how many in a formal sense, it is a measure of the number of microstates that you have available for you to explore. Okay? So in some sense, how many different configurations can you take that are consistent with the overall macroscopic properties of your system, like overall energy, overall temperature, overall pressure, and so on. Okay? So keeping these macroscopic things fixed, how many microstates, how many conformations can your DNA access? Now, you'll realize that when you have this open state, you can access many more conformations. You can actually count, for example, using simple models, as I'll show in subsequent lectures, you can count how many conformations that you have accessible to you for an open conformation like this versus a very condensed conformation over here. And the thing with these condensed conformations is that you do not have a lot of configurations that you can explore. So, for example, let me take a segment of the polymer over here. So let's say this polymer is corresponding. Let me draw using a different color. So let's say I consider this segment of the polymer over here in the condensed state, which in this open state is something like over here. Okay, this is the corresponding section. Now, this polymer, this segment, so for example, can explore, it can perturb a little bit along this direction. Maybe it can perturb a little bit along this direction. Remember, this is very simply, I'm just trying to give a very diagrammatic sort of feel for this entropic argument. We can use more concrete arguments, and that we do in class, but I'm just trying to give you a very qualitative feel for what is going on. Okay? So let's say I have these three conformations that are available to me. Okay? One is this original one, which is in this blue line over here, and then it could go maybe a little bit out in this direction, or it could go a little bit out in that direction. Okay? So I have these three conformations that are available to me. Okay? And if you know, so these are the three possible microstates in this very simplistic picture. And if you know, like Boltzmann's formula, how to calculate entropy from microstates, you know, Boltzmann's famous formula is that S is like kb log of the number of microstates. Basically, it's proportional to the number of log of the number of microstates. So here you have, let's say, three microstates that are possible. One is this original, and then there are these two perturbations that I have drawn, one on this side, one on that side. Okay, so you have some entropy which is like log 3. Now let's look at this condensed state on this side. Okay, now here is my original conformation. Now I could move it, let's say, over this direction. Let me first draw it so that so here I could perturb it something like this along that direction. Okay, so I give it a slight kick in this direction, and that's allowed. On the other hand, if I try to give it a slight kick in this other direction, I come across this other strand of the DNA that is already present in that space. Okay? So that confirmation is no longer available to me. So in that sense, the number of confirmations that I can access in this very simple picture, here I had, in this left-hand side, I had 3 confirmations that I could access. Here I have only 2 confirmations that I can access, because the third one is blocked, because there is already some DNA, already some polymer that is present where I would like to go. So therefore, what that means is that effectively I have a reduced number of microstates that are available to me. If I have a reduced number of microstates that are available to me, I have a reduced entropy, okay? And therefore, these structures, so these supercoiled or sort of condensed structures, undergoes a reduction. So the entropy in this state is greater than the entropy in this state. So this entropy in this open state is greater than the entropy in this closed state. So then what drives these sort of things? It could be sort of energetic, it could be energetic considerations. So for example, if you have proteins that are binding, charged proteins maybe, which bind and which sort of bring together different segments of the DNA, then it could lead to collapse. Depending on solvent quality, you could have collapses and so on. So there are different things that lead to transitions from this open state to the supercoiled state. In biological situations, it's mostly interactions with different sorts of proteins. But if you were to look at purely an entropic argument, just this open state versus closed state without thinking about protein interactions and so on, what this very naive sort of entropy estimate says is that the entropy of this open state is going to be greater than the entropy of this closed state. All right. Like I said, so when we do these sections on polymers, we'll deal with this a little more formally. So we'll see how to write down the free energies of polymers explicitly, and from there, how does one calculate? So therefore, what that means for entropy. Polymers as entropic springs is in fact something that I cover in some detail. Okay, so. This is a very hand-waving sort of an explanation. Once I covered this topic in the subsequent weeks, if it's still not clear, you still have doubts, please feel free to ask, and I can again address this in one of these future live sessions. Okay. Yeah. So also please let me know. So since this is the first live session of the course. We fixed it at some time. If sometime, please let me know, what sort of frequency you would prefer. For example, if you want me to come online once every 2 weeks or so, I'm happy to do that and clarify whatever doubts that you have. If a different time is sort of more suitable, again, please let me know, and I'll try to make accommodations for that, depending on my schedule and of course the NPTEL schedule as to what is available. But so basically, I'm sort of flexible. What I hope is that if you have a doubt, it's best you ask it to me, because you're doing this course, which is very nice, and you're doing it because you're interested in it, which is also very nice. So therefore, since you're taking the time and effort to do this, you should put in the effort to do it well, and I'm happy to help you in that in whatever way I can. Right. So please ask more questions on the forum. If you want more live sessions where you can directly ask more questions, again, please feel free to do that. I'm happy to come online more often, even if it's like shorter sessions. They can just come online for 20 minutes, half an hour, and so on, and try to answer some questions that you might have. If there are topics, that I'm not covering in a lot of detail, but still you feel you're interested in and you would want to know a little bit more, again, you can feel free to ask if I can. So if that is something I know a little bit about, I'm happy enough to come online and talk about it as far as I know. it need not be part of the sort of examinations or assignments or something, but it's just for like a learning purpose. Okay, so that's also fine by me. All right. What else? Yeah, I think. Are there any more questions on the YouTube chat? I don't think so. Are there more? Yeah, I think I've sort of covered more or less the questions. So if you have any further things, you can just type in the chat box. If not, yeah. So if not, we can end this session here, and we can meet back periodically every couple of weeks or so. So there was something else I wanted to say. Yeah, so I think there's also some exam registration deadline. So if you have not done it yet, um, I think you should do it as soon as possible. Yeah, I have to check the deadline, but please try to make sure that you do it before the deadline. We'll try to cover. So the way this course sort of works is that there are a lot of these different things that I take up and I sort of look at in a little bit of detail. Not too great detail. Um, but enough to sort of give you a flavor of the topic. My aim is basically that biological physics or biophysics is a very broad subject. It's a very new subject in some sense. There is a lot of scope for young students or anyone, professionals, researchers, to make some research contribution in this field. So unlike in some older, more established fields, where a lot of things have already been done, but biophysics, or at least theoretical biology, in that sense, that I work on, is relatively. I mean, it has a certain history, but it has sort of taken off in earnest over the last 20, 30 years or so. So there are a lot of questions in biology where there is really a need for collaborations between experimental biologists, so proper people, biologists who do experiments, and people who are not really biologists by training, but who are willing to talk to biologists in order to understand that what are the questions that are involved. So whether they be physicists, whether they be mathematicians, whether they be engineers, all of us can, depending on what your skills are, you can, if you're interested, if you find biology fascinating, and you should. I mean, if you think about it, uh, it's an extremely complex system, which we really, so even a single cell, if you think about it, is an extremely complex system, which we do not really understand how the different components of that, all of them, fit together to work in such a reliable fashion. So there are a lot of open questions, and depending on what your skill set is, whether you are a physicist, whether you're an engineering student, or whether you're a mathematician, whether you're a chemist, you can bring a certain new way of looking at a problem. Okay? uh, Depending on what your training is, depending on what your interests are. And by bringing this new way of thinking about a problem, you can actually contribute to understanding certain fundamental things about biological systems. Uh, so that's sort of what I try to give a flavor of in this course. I do it through tools that I'm comfortable with. But if you find these systems interesting, if you find this sort of biology as a topic, how life emerges out of these if you think about it, very simple physical and chemical processes, some sort of reactions are going on. There are some mechanical forces, but out of these apparently simple processes emerges something as complex as life. it's a very challenging question. It's a very fascinating question. And if you think that this is if you find this as fascinating as I find it, there are ways in which you can contribute to this field, regardless of what your background is. So that's sort of the feel that I want to give through this course. So hopefully, you'll stick with this course through the end. Continue asking questions on the forum. Continue asking questions for live sessions in the future. Let me know, if I should focus more on some things versus others, and I'll try to you know do as much as I can within the constraints of this online system. Okay, all right. So I'll end this live session here today. Best of luck. Hope the course going forward is also interesting. And please give me your feedbacks. Continue to give me your feedbacks as more and more topics are covered, so that I can do a little better as well. All right. Thank you, everyone, whoever is listening. So thanks for coming, and yeah, bye. All right. Thank you.